I believe we now have our second round of poster pitches.
Hello, my name is Christine, and I'm a PhD student under Associate Professor Elena Papaleo at the Technical University of Denmark and the Danish Cancer Society Research Center. And today I brought this poster. And the poster describes some of our research by illustrating a workflow for studying the impact of mutations in protein structures in silico. And for this purpose, we have worked with the known cancer mutations of tumor protein 53. Effectively, we have simulated over 1,000 mutations on the stability of the protein, the effects on local binding sites, and the effect on distal sites via allosteric pathways. And to do so, we have applied a broad scope of computational tools and approaches, and we have been able to predict conformational changes as a consequence of these mutations. And I really hope to get to talk to a lot of you about it.
Hi, I'm Bing, a grad student in Dr. Craig Kaplan's lab. How do protein residues interact with each other to ensure proper functions? With an essential domain of e-RNA polymerase II, the trigger loop, as an example, my project dissects almost all residue interactions in the trigger loop by a highly reproducible deep mutagenesis structural genetics system. We identified functionally dependent residues in the trigger loop. Coupling analysis suggests those dependent residues may be co-evolved. The functional interactions we identified in the trigger loop are important for understanding RNA polymerase mechanisms, and most importantly, our highly reproducible deep mutagenesis system for detecting residue interactions is applicable to any other protein. Thanks. Please come back to my poster.
Hi, my name is Alberto Escobedo, and I'm a postdoc at Ben Lenner's lab at the CRG in Barcelona. The main goal of my postdoc project is to build accurate predictors of protein activity from sequence information alone. To do that, we measure comprehensive maps of the energetics of the interactions taking place in protein cores to build protein stability predictors, and in binding interfaces to build binding affinity predictors. In the first part of the project, we are combinatorially mutagenizing protein cores, in this case, the SH3 domain of the GRB2 protein, where we have found an alternative core configuration, which is 7 mutations apart from the wild type. In the second part of the project, we are combinatorially mutagenizing several positions in the leucine zippers and all the contacting residues, and we have been able to infer the energetics of mutational effects and also pairwise epistatic interactions that we have used to build highly accurate predictors for binding affinity.
Hello, my name is Clayton Friedman, and I'm a postdoc in Dr. Daniel Yang and Chuck Murray's groups at the University of Washington. And today I'm sharing ongoing work in a collaboration with Leah Stravita and Doug Fowler's groups, where we are developing a strategy to be able to execute DMS and MAVES in human-induced pluripotent stem cells and their derivatives. And in particular, my work is focusing on mutagenizing myosin-7, which encodes a sarcomeric protein important for cardiomyocyte function and is a common cause of hypertrophic cardiomyopathy. And we developed some pretty slick strategies to be able to generate these libraries in plasmids, mutagenize iPSCs, and then differentiate the cardios and put them through a pretty standard growth assay. So if you want to learn more about the results, come find me at the poster in GatherTown.
Hello, Toronto. My name is Andrea, and I am a data scientist from SEM, the Research Center for Molecular Medicine of the Austrian Academy of Sciences. With my poster, I would like to introduce you to Resolution, a project founded by AMI, EU, and EFPA, where partners from academia and industry are working together to study the impact of genetic variants in solute carrier transporters. Our goal is to exploit state-of-the-art experimental and computational technologies, such as the deep mutational scanning and the AI variant effect predictors, to understand which variant can lead to the development of diseases in humans, the molecular mechanism behind this, and ultimately find novel therapeutic targets. I hope I have piqued your interest. If you have any questions, please reach out to me during the poster session, and see you soon.
Hello, my name is Renee Geck, and I'm a postdoc in the Denim Lab at the University of Washington, and I'm conducting deep mutational scans of human G6PD variants in yeast. I express G6PD variants in yeast and then grow them in continuous culture under oxidative stress, so that over time, variants that are known to have low activity decrease in frequency in the population, and variants that have wild-type-like activity increase in frequency. I've used this system to conduct a deep mutational scan of over 10,000 G6PD variants. And from preliminary analysis, I can see that important functional regions like the dimerization domain are enriched for variants that have decreased functional scores. I can also see that variants that are known to contribute to G6PD deficiency have lower functional scores than synonymous variants. I'd love to talk to you at the conference, at the poster session, or over email if you'd like to learn more about this project or other ongoing work that I have on interpreting variants using both clinical and functional data, as well as exploring the effects of background haplotypes on variant function.
Hi, I'm Sarah Gassing, and I'm a PhD student at the Hartmann-Petersen group at the University of Copenhagen. My project is focused on the human enzyme glucokinase. Glucokinase regulates insulin secretion in response to changes in blood glucose levels. The activity of glucokinase is finely tuned to maintain glucose homeostasis, and variants that decrease or increase activity are therefore linked to several diseases. Loss of function variants can cause glucokinase modi, a monogenic form of diabetes. Patients with glucokinase modi are frequently misdiagnosed and receive unnecessary treatment. In order to promote variant interpretation and diagnosis, we generated a variant activity map of glucokinase. We scored both loss and gain of function variants and used computational predictions of protein stability to examine potential variant mechanisms. If you're interested in hearing more, please come find me at my poster.